My name is Nikki. For those of you who don't know me, I'm based at Forest Research, and I'll be presenting alongside my colleague Guillermo from Kew Gardens. As we are all aware, there is a global initiative established to expand forest cover as a means of safeguarding biodiversity and mitigating climate change. Tree planting has become a widely adopted strategy to achieve this goal, supporting the natural processes of forest regeneration. We were interested to see if tree populations of English oak and silver birch were being represented by a diverse range of seed sources, as we believe that that will improve the genetic diversity within populations, in turn perhaps improving tree susceptibility to pests, disease and climate change. If there is a decline in genetic diversity and inbreeding occurs, this could lead to potentially unsuitable tree species to help with the threats they are likely to be facing in the future. So the aim was to compare the levels of genetic diversity in planted versus naturally colonised forests using a lot of genetic data to try and identify potential population bottlenecks by loss of genetic diversity and or selection across age cohorts, whilst also considering differences in health indicators using statistic modelling. So I'll spend a few minutes just going over the project and methods, etc., but I wanted to outline some of the summary findings. So we will revert back to these findings at the end of our presentation, but I wanted to go over what we'd summarised. So we initially found, based on information obtained from forest reproductive materials, it seems that for more than half of the 39 UK tree species analysed, seed collection appears to significantly underrepresent the range of climates in which those trees naturally occur. This probably needs to be taken with a pinch of salt, as it might not include all the stands and might have correct geographic locations, but it's what we found from our initial investigation. We also found that overall genetic diversity metrics for planted and naturally colonised are quite similar, which is good news. It means that the planted populations can emulate the genetic diversity of wild populations. However, on the other side of this, the planted populations were more similar to one another than, say, a naturally colonised set, particularly for oak. We also find weak but significant evidence that there is selection in nursery pipelines, so we may or may not be introducing biases to the cultivated material. So what we're looking to compare. So current afforestation strategies in the UK to meet conservation goals can be broadly classified into two main types. So afforestation pipelines based on natural colonisation or regeneration, where tree communities establish in, say, former arable or farming lands by natural dispersal. And then we have the afforestation pipelines based on active planting using commercially produced trees. So each strategy involves different processes that may affect the levels of genetic diversity in resulting woodlands. So parental trees in wild populations, for example, may arguably represent a wider genetic pool and be better adapted to the target site than those in geographically distant seed sources or orchards. But in turn, selective pressures across age cohorts, such as climate change, predation, pests, diseases, may reduce genetic diversity through differential mortality in natural populations. Conversely, planting protocols that integrate multiple seed provenances and aim to maximise germination and survival through highly favourable growing conditions can result in more diverse populations than natural ones, in theory. It is possible that these populations, through natural selection, are better to keep pace with emerging stressors. However, commercial seed collection, germination, nursery cultivation and or planting protocols can also potentially inadvertently introduce genetic bottlenecks or selection at one or more of the life stages. So in this study, we focus on 3 different age cohorts across the natural colonisation and planting pipelines to test these competing hypotheses. So sites were selected based on their planting and colonisation of the target species. So we were interested in oak and birch, and we did this on former arable land, with existing woodland located nearby with mature trees that are seeding and therefore the potential trees for the colonisation. It did prove to be somewhat difficult, as we know birch seeds from such a young age. We obviously didn't want the natural colonisation to be seeded from the planted populations. Without going into too much detail, I'll hand over to Guillermo now, who will give an overview of the results from the genetic side.
Thank you, Nikki. I hope that you can all hear me well. So yeah, as Nikki already mentioned, and before analyzing genetic differences between oak and birch afforestation strategies, our first aim was to test if the variability in environmental conditions of the tree populations in the wild were sufficiently represented by seed sources. So our aim here, the idea behind this test is that if different wild populations are adapted to different conditions, but we rely on only a few of them as seed sources, we can lose some of this adaptive variability in planted populations. To understand these potential environmental differences between wild populations and seed sources, we first mapped registered seed sources stands obtained from the UK Register of Basic Materials, as well as from master certificates when they were available, for a set of 39 UK native species. You can see some of them as an example on the left panel of the slide. The second thing that we did was to extract climatic values for all the variables listed on the right at the seed sources for each one of the species, as well as across their wild distribution. And then we tested for significant differences between these two sets of data. So what we have here on the left, what you can see here is a spatial representation of the climatic condition of seed sources in green and of the climatic range of wild populations in orange, based on a principal components analysis. A principal components analysis is basically an approach to summarize variability in a large data set with many variables to be plotted into a bidimensional plot like you have here in the slide. So this is not a geographical map. Each dot represents an individual or a data point and occupies a location in the plot depending on environmental scores measured where they occur, yes? So the important thing here is to notice how the wild populations consistently cover a broader range of conditions than the seed sources. And this was the case for 23 out of the 39 tested species. These differences were significant in 23 of all the species tested, including the two species of interest in this study, Betula pendula and Quercus robur. So this suggests that seed collection often overlooks the full environmental variability found in the wild, which may reduce the adaptive potential and resilience of planted populations to current climatic conditions and also those changing in the future. It is important to note, though, that this analysis is based on the UK Register of Basic Materials, which likely doesn't include all the stands used for seed collection. With more complete records, we should be able to test this pattern in greater detail and with higher accuracy. Okay, so moving on to the genetic analysis on oak and silver birch, on pedunculate oak and silver birch. Let me tell you a little bit about our sampling design. We sampled around 25 individuals per cohort. The cohorts that Nikki already mentioned, parental trees, seedlings, and saplings, from 4 wild populations where natural colonization was ongoing. For each one of the species, you can see the locations in the map in the slide. For planting pipelines, we sampled trees from 3 planted sites, also shown in this map. We sampled also seedlings from different origins, from 2 nurseries, Mylord Forest and Cheviot trees. And in the case of birch, we could also sample a seed orchard. To complete the data set, we also included samples from natural stands representing seed sources. And in total, we obtained genetic data for 459 oaks and 625 birch trees. So with this data, the main questions that we wanted to answer here were, first of all, is genetic diversity in planted pipelines higher or lower than naturally colonized populations? So we have 2 competing hypotheses here that need to be tested. In the first scenario, seeds sourced from a limited number of parent trees can result in populations, planted populations that have lower diversity compared to wild ones. But also, on the contrary, seeds sourced from multiple provenances and mixed may actually result in populations that have higher diversity than reference wild populations because of this multiplicity of origins. In second place, we wanted to test if planting pipelines can relieve inbreeding compared to naturally colonized populations. Small and fragmented natural populations may be at risk of inbreeding and genetic drift, and planting pipelines done in a particular way may actually help to reduce this inbreeding in afforested populations in theory. Third, we wanted to test if planting pipelines generate sites with more similar genetic backgrounds than natural ones, or in other words, we wanted to test if planted populations were more similar, genetically speaking, to each other than natural ones. And this could happen through different processes. On the first scenario, using the same seed sources and multiple sites can obviously generate homogeneous planted populations, right? But using seed mixes, using the same seed mix in multiple sites can also generate populations that are more similar to each other, even if they are more diverse, as shown in the figure. The opposite scenario will be using site-appropriate seed sources, which can help to maintain population differences. And finally, we wanted to test if planted pipelines were introducing biases, were introducing selective pressures through the process. Seed source selection, nursery, cultivation, and planting can actually introduce these biases, this selection, which is something that we can also test with genetic data. We started by testing for differences in genetic diversity using several indices. So each one of these symbols here in the y-axis are basically different ways of measuring genetic diversity. Each bar represents a population or group within each cohort or life stage shown above the bars, and for each one of the afforestation pipelines. So as you can see here, we found very minor differences when comparing groups corresponding to natural colonization and planting afforestation pipelines. The differences were non-significant for almost all of these comparisons. So what we can infer from this in regard of our first question of interest is that first, we don't have lower diversity due to limited seed sources compared to wild population. And second, we don't observe either higher levels of genetic diversity in planted populations as a result of mixing seed origins or because of variable conditions in the first stages of their life. Next, we tested for inbreeding across cohorts in both wild and commercially produced trees. Inbreeding measures the potential genetic loss, of genetic diversity as a result of mating between related individuals. Using a statistic called FRO, we found that most wild and planted populations had similar levels of inbreeding, except for natural colonizers. This is the seedlings and saplings that we sampled in recently colonized former arable lands. So on the plot, what you can see here is that the populations corresponding to the planting pipelines had low levels of inbreeding, as well as adult trees in the natural colonization sites. On the contrary, we had the colonizers of these arable lands, former arable lands, showing higher levels of inbreeding, and this pattern was repeated for both species. The truth is that during fieldwork, Nikki and I often observed that adult parental trees in natural colonization sites occur in small isolated patches. In some locations, I was even able to sample every neighboring adult tree because they were so few. This is so unusual that I was able to sample every individual in a population. So this reflects how high is the risk of inbreeding in these populations. These results suggest that the planting pipelines can indeed effectively prevent inbreeding in afforestation programs, especially when seeds are sourced from orchard or from large mature forest populations rather than these small fragmented stands that we saw in natural colonization sites. Okay, next, we tested whether planted populations were genetically more similar to each other than natural populations, and we did so by calculating co-ancestry scores, which measure how closely related individual trees are. High co-ancestry is not the same thing as inbreeding that we explored in the previous slide. Inbreeding essentially estimates how closely related an individual's parents are, while co-ancestry basically measures how closely related I am, for example, to somebody in my populations of individuals. So the key connection between these two concepts is that a population with high co-ancestry is always at risk of producing inbred offspring in the subsequent generations. And that's the risk that we are measuring here. Co-ancestry scores can be calculated within a single population or between different populations. The results are, for this analysis, are shown in these two heat maps for oak and birch. So let me explain a little bit about how these plots work. What we have here, so it's basically the same populations or group in rows and columns. So each cell represents a comparison in between two of them. Along the diagonal, we have a comparison of each group with itself. So the diagonal is a within group co-ancestry score, and the remaining cells are the pairwise comparisons. Let me remind you a little bit of the structure of our dataset here. We have 4 natural colonization sites for each one of the species with 3 different cohorts: adults, seedlings, and saplings, colonizers. And then we have the populations or groups corresponding to the planting pipeline. We have seed sources, an orchard in the case of birch, nurseries, and outplanted populations. There is a lot of information in this plot, so let me guide you through the key findings here. The first pattern that we observe is that the highest levels of within group co-ancestry occur in the populations or group corresponding to the planting pipelines. So we have these cases here of nurseries and outplanted populations in oak, and basically almost every group in commercially produced trees in the case of birch. And this is problematic, right? Because as I already mentioned, high co-ancestry can result in inbreeding in subsequent generation, and that can also result in a loss of genetic diversity. There was an exception to this pattern of heightened co-ancestry in commercially produced trees, which was the only orchard that we had in our dataset. And this is encouraging because this suggests that orchards can be a good strategy to avoid this problem, this risk of high co-ancestry and subsequent inbreeding. The second pattern that we observed is that co-ancestry scores among populations in wild populations in natural colonization sites, basically those comparisons within that triangle, were lower in natural populations compared to comparisons between or among nurseries and outplanted trees, which are represented within the lower triangle. So this indicates that there is, to some extent, to some degree, some genetic homogenization going on in the case of oak. And this is probably due to the fact that the vast majority of seeds used in the afforestation of oak in our dataset came from a single seed zone, the zone 405. We didn't find this pattern, or at least not so pronounced, in the case of birch when we compared the two groups. And this is most likely because in the case of birch, there was way many more origins of seeds used for planting, which helped, it looks like, to maintain genetic differences among populations. So in summary, planted pipelines can indeed lead to more similar genetic backgrounds as we see in oak. However, using diverse seed sources, as in birch, can help preserve genetic differences between populations. Such is the goal, for example, of favoring local provenancing. We could not test if seed mixing can also result in more similar populations because we only had one such population deriving from the birch orchard, so comparisons couldn't be made. But at least we found this pattern, this result of absence of heightened co-ancestry scores with respect to wild populations. So the last question using genetic data that we wanted to answer here was whether selective processes are occurring in planting pipelines. For this, we focused only on colonizers and outplanted trees. We excluded adult trees and those from nurseries because we wanted to see if being planted or naturally colonized explain to some extent genetic differences that could occur between these two groups using a statistical method that is known as redundancy analysis. So to illustrate this analysis, think of a group of people from which we collected height measurements. If we group them by nationality, let's say these people are just Dutch and Spanish, we will most likely see a significant average difference, as the Dutch are the tallest people in the world on average. But if we classify them based on a less relevant variable, like a preference for the beach or the mountains, we will probably not see a meaningful difference between the two groups because this variable is most likely not informative of height, okay? So here, our explanatory variable is planted or not, and instead of heights, we have genetic profiles, we have genotypes. As these plot results show for oak and birch, colonizers in orange and planted trees in blue do not overlap. They do show differences. And this clear separation between groups indicates that the variable planted or not is indeed relevant. Or in other words, this indicates that being a colonizer or a planted tree is associated with genetic differences, which in our analysis were limited but significant. This analysis also allows us to identify which specific genes along the genome show greater differentiation. In our test, some of these genes had functions particularly relevant, such as genes influencing flowering time or for how long seeds stay attached to the tree. These are traits that could be unintentionally selected during seed collection, for example. So, do planting pipelines show evidence of selection? Yes, we found a clear signal of selection. However, further research is needed to understand the strength of these signals and their effects on the resilience of planted forests, because as it is at the moment, we ignore if and to what extent these potential biases in planting pipelines, in planting protocols, can impact the sustainability of resulting populations. Nikki will now present data on health indicators, on pest incidents, which also suggests that planted trees and colonizers may be facing different selective pressures.
So yeah, so not only did we look at the genetics, but we also considered the phenology of the naturally colonized and planted trees. We were interested in assessing the main pests and diseases associated with birch and oak, so such as oak powdery mildew, leaf spot on birch, galls, leaf rollers, crown health or crown dieback. We also visited the trees in winter to assess their form, as indicated in the diagram on the right-hand side. All of these variables that we assessed are associated with causing the tree some form of stress by either affecting its photosynthetic capabilities or causing stunted growth, crown dieback and such. So what did we find? Overall, the trees are actually quite healthy, so that's great news. There were few differences observed between planted and naturally colonized. Now, we can surmise that as most of the trees at the sites where comparisons were made, the youngest were already 10 years old, +2 for the planted ones that were grown in the nursery initially. As we're comparing trees of the same age for health assessment, they are probably past an age where they are considered to be vulnerable. But obviously, this was just the sites that we could find and the age of trees that were there. Most observations were visually recorded, so we also performed an assessment via chlorophyll fluorescence, and this also supported the visual protocols that the trees were found to be in good health. Statistically, we did find that there is a lower likelihood of mildew in planted oak, and there is a higher likelihood of leaf spots in planted birch associated with the fungus Mycosynina betulae. Obviously, I went over the summary results at the start, so we're going to go back to those now. So as Guillermo went through, for more than half of the 39 UK tree species analysed, commercial seed collection significantly underrepresents the range of climates in which those trees naturally occur. So, for instance, we're collecting seeds from Seed Zone in Scotland and then planting in the south. We don't fully understand the impacts of doing this. The trees all appear to be healthy, but should we focus more on where the seeds are being sourced? We record the seed zone so that we can look at the provenance, but what about specifically planted trees from, say, Seed Zone 405, which is where the majority of our oak was from, on the same plane as the target planting site? So there are lots of questions that we still want to answer and wanted to discuss. Could we increase the number of parent trees? Seed zones are quite big if you look at the FRM map. Can we get this more knuckled down to what 3 words or GPS coordinates perhaps? If we only use registered stands, are we then limited? Do we need to get more stands and increase the genetic variability. Obviously, we don't want to make the processes more complicated. Nurseries find it as complicated as it is. So how can we do all of this to make it easier for seed suppliers and nurseries? As Guillermo said, the overall genetic diversity metrics for planted and naturally colonised are quite similar, which is amazing news. But there was evidence to suggest inbreeding in naturally colonised seedlings and saplings, so potentially due to a limited number of adult seeding trees, which highlights the importance of planting. But what about the naturally colonised trees? If we aren't using suitable planted trees, then what could happen in the future with the trees that have naturally colonised with limited genetic diversity, perhaps? Again, just something to think about. By contrast, co-ancestry showed that planted sites were more similar to each other than a similar set of naturally colonised sites. This was in particular for oak. Well, what does this mean? So basically our planted sites in the east are therefore genetically fairly similar to those in the south. So the same trees are being used for seed sources, which makes sense when you think about it. Nurseries outsource a lot of seed collection, unless they have their own orchards. And if you were collecting seed and you knew where the best seeding trees were and when, you would go back to them. You wouldn't waste time looking for new trees. But this means genetically our planted trees across the UK could be quite similar. And we don't know what impact this will have in the future. It might be fine. We don't know. This is obviously a sensitive subject as well. It's difficult to approach because obviously we don't want to say that one person's doing it right or wrong. With a fairly limited number of seed suppliers in the UK, how could this be improved moving forwards? We find significant but weak selection in the nursery pipelines. Again, this is understandable, but if we are selecting saplings for planting that are, say, for example, all the same height or of a certain root collar diameter, this could lead to a selection of functional traits associated with flowering time and acorn detachment, for example, which might not therefore be adapted to where they're being planted. Obviously, thinking of a worst case scenario. There might be a loss of genetic variability or functional traits that could be useful in the future. Again, with birch, we found the number of planted trees to have significantly higher infection rate with the leaf spot, although this could be due to a result of being planted next to each other and the stand density is much, when they're much closer. So everybody that's been involved, there's been so many amazing people involved in this project, and this isn't even half of them. We've really appreciated everybody's help. I know some of you are here today, got some private sites as well that, you know, we're very open to letting us go on and do lots of sampling. We did do the phenology over 2 years as well. So we went and visited them 2 summers in a row and 2 winters. And yeah, everybody that's been involved has just been amazing. So a huge thank you to everyone and whoever's here today. And I can see there's lots of questions, which Louise, do you want to mediate?
Yeah, yeah. Thank you so much for that, both of you. That's fantastic. Yeah, we have got quite a few questions. So very first one is from Alex Pritchard, who said, in your investigation, is there an increased trend in hybridization of Quercus petraea and Quercus robur? This can be seen in a number of oaks close to Rydal in the Lake District, and could this be provenance related?
Actually, maybe Louise, you are better equipped to answer this, right? Based on your investigation. The thing is that because it's so easy for Petraea and robur to hybridize, we used in our analysis a reference genome that is a hybrid. So even when we try to target a more consistently Quercus robur, there may be some hybrids or even Petraea in our data set. There may be the least. But we don't look into patterns related with hybridization. So I cannot really comment a lot on that. So maybe Louise, you can actually
Yeah, so I'll add to
knowledge.
that, Alex, and do feel free to contact me if you want to, because I did part of my PhD on hybridization, and where you are in the Lake District is an area where natural hybridization is very common. And so it's more likely to be natural than provenance related, I would think, but I'm happy to talk to you about that offline at some point. A question from Joe Clark. So, Guillermo, I think you mentioned that there was a sort of limited number of parent trees, and she's just saying how many would you consider to be a limited number?
Well, the thing is that we say that there may be a limited number of parental trees based on our data, based on our data set, which obviously it's very difficult to capture all the ongoing variation going on in all the plantings, but based on what we saw. We say that there is a limited number of parental trees compared to wild populations. That's our reference. So if I had to go for a number, I couldn't say a number of trees particularly, but something that actually reflects or emulates what we see in natural populations. We infer that there is a reduced number potentially in the case of oak of parental stands, basically because the co-ancestry among planted population is higher when we compare planted populations with each other than when we compare natural populations. So there is, I mean, based even in our results, it's difficult to make a specific recommendation in that sense, because if you favor, for example, local provenance, and if we limit our seed collections for planting to a seed zone, obviously you have more chances that you're going to have higher co-ancestry in that group, right? I guess it can even be, there's room for improvement possibly even within a single local provenance. In the case of UK, and this may be a bit well far-fetched, but the thing is that we've seen in many different species in the UK of trees that the population structure is relatively low or low. So maybe in the case of the island of Great Britain, the local provenance in favoring local provenance can be not that favoring, not that positive, and including seeds from further regions, if that results in lower levels of co-ancestry, that could be actually a right way to move ahead. But even within a single seed provenance, for example, the seedable co-ancestry patterns could be at least those that are similar to what we find in the wild.
Thanks, Guillermo. And another a question from Annika Perry. What age was a sapling?
So the thing is that in our first design, when we were being very, very optimistic, we wanted to separate the seedlings and saplings based on basically rules of thumbs to classify seedlings and saplings, which I don't quite remember now, but it has to be with a particular DBH and a particular height, right? But we realized that it was impossible to actually make to actually build similar groups in terms of numbers to be compared. So in each population, basically, we divided each set of colonizers in two based on DBH and height, but they're basically split in two based on those two metrics. So planting, well, the year in which these former arable lands were left to regenerate ranged from 2011 to 2019, if I recall well, Nikki. Maybe you know about this better than me. But yeah, we have different ranges of age, different ranges of classification into seedlings and saplings.
Yeah, thanks. So I think the reason for that was Annika said it would be interesting to also test trees which are reaching reproductive maturity, as you might expect the inbreeding coefficient to reduce as the trees age due to fitness costs for inbred individuals.
Yeah, at some point it will be awesome to be. There was we had some problem. So our idea here was to be able to compare stages along planting pipelines against what we see in the wild, right? So we had in planting pipelines, we have seed sources, we had nursery trees, and we had outplanted trees in their early years. We could have gone further than that, but we selected those three categories or age classes so we could compare the two afforestation pipelines, basically. But yeah, looking forward into adult planted trees or adult regenerated trees, that would be That would be nice to do.
Yes. Yeah, so the nature of the sites meant that that wasn't feasible. One, Nikki, you might be able to answer. The FRM requires an Ordnance Survey grid reference for every collection, and could you use this to refine seed source information?
Yes, but obviously we were limited into what we could access, and we're also relying upon that being correct. I mean, dependent on the seed collectors, they're just collecting from a certain region and just giving a coordinate, but it could be from like 20 miles away. It's not as accurate as we hoped it would be. So, but how we're supposed to moderate this, how anybody is supposed to find that exact information out is one for the future, I think. But
Thanks,
yeah.
Nikki. Derman says, this is probably for you, Guillermo. Wouldn't you expect that levels of inbreeding in naturally colonised areas, especially fragmented ones, are to be expected and therefore not considered problematic?
The truth is that we didn't expect it. This was a result that we just found, and we thought it was so interesting. Yes, I guess if I had thought about it before actually running the test for this particular result, I could have guessed, but we were possibly more inclined to think that maybe bottlenecks in planting pylons could result in inbreeding. The truth is that this statistic, and actually every statistic that measures for inbreeding, can also be a proxy for genetic drift, which is not exactly the same process. Genetic drift affects small populations in which variability is lost due to random processes that don't really occur, or not so seriously, when the populations are big, because the alleles are more easily maintained in big populations. So I guess if I had, yeah, we could have a hypothesized from the very beginning that these small patches surrounded by former arable lands or still agricultural lands could result in this pattern. That said, I mean, that doesn't make it less problematic in the sense of if we're going to relate in natural colonization for seeding, that could be positive if seeding populations, the populations of adult trees in this natural colonization site, were large mature trees, but that's not the reality of the landscape, right? Not in countries like the UK or any country, for example, with this level of development across the landscape. So I think it can be actually problematic in the future if we only relied on that, and also like targeted planting, assisted gene flow can help to prevent this.
Thank you. So a follow-on question from me on that, and I can see Jeanette Hall has also put a comment on this. But so bearing in mind that both the species you're looking at are sort of quite widely distributed and they're wind pollinated, and we know from other studies that there isn't a lot of population structure in them. Do you think there's something really specific about these agricultural sites that might make the results different from a more continuous distribution of woodland if your samples were from elsewhere?
You mean in terms of the trees that are established in those areas, or?
Yeah, in terms of the inbreeding scores, you know, because these sites are perhaps more fragmented than other sites where these trees might be found.
Yeah, no, obviously, I mean, this is, again, I wouldn't say speculating because the results are kind of clear, but this is our hypothesis about it is that inbreeding is due to the small size of these populations that are sitting these areas, and if regeneration or natural colonization happens in other areas that are more afforested, where seeding forests are larger and more mature and not subject to drift, maybe we wouldn't see this pattern. But the truth is that this fragmented setting of seeding trees was predominant in all the populations that we visited, and we visited a lot of them. Not only the ones that we are showing here, I think we visited between Nikki and me nearly 50-55 populations. Some of them were discarded because of a number of reasons that didn't fit our requirements. But in terms of what kind of land use from before, we suspected that there can be an influence, and that's why, to control for those potential differences, we focus only on one type of land. That doesn't mean that there are no differences. We found difference in terms of a slope, of drainage, but at least we wanted to control for that. So potentially yes, but we don't know really if that implies a difference in the trees that get established. Also in terms of inbreeding.
Thanks, Guillermo. Right, next question. Fraser Bradbury says it's interesting to see the limited seed sources for oak in the UK. Do you know how the introduction of European source trees affected the genetic diversity, as we've been using European seed sources for decades, if not hundreds of years? And yeah, the seed sources from anywhere it can be collected if there are poor mastering years.
Well, we actively avoided populations that were planted with seeds that were not from the UK, because that's what we wanted to test. And that was also very limiting, because in many cases it was very, very difficult to track down the origin of the seeds. That was Nikki's work, really. She did an amazing job on that. So no, we don't have data on how the use of seeds from outside the UK, from mainland Europe, may affect the diversity in plant population. But it's true. I mean, the landscape, the forests are so transformed. There's been planting for many years, centuries even. So there is most likely a signal of that even in our data set, but we didn't test for that particularly.
Okay, thank you. Jonathan Norris asks, as there's no major difference in genetic diversity between planting and natural regeneration, does this bode well for adaptation to climate change? I.e., whether we want to plant from 3 degrees south or locally adapted natural regeneration, they both stand an equal chance at successfully adapting?
Well, that's something that we may be able to answer better in the next phase of our project. We just got an extension to test, similar to what Phoebe was showing before, these levels of maladaptation, potential maladaptation, or genomic offset, when basically you compare the correlation between genotypes and environmental variables. From what we see, and also this is something that came out in our previous meetings with the stakeholders that we had in the previous few weeks, there is a sense of that there is not so much phenotypic variation latitudinally, although Romulo and Dolmen just show the opposite. In terms of how diverse planted populations are, I would say that I can only speculate about how beneficial may be to actually loosen or relax those restrictions, and maybe allow for more seed mixing, for planting seeds from other seed zones. The seed zonation system itself, it's maybe not super informative in terms of environmental variability. So maybe there are better ways, and maybe this 3 degrees latitude rule is better than, for example, seed zonation for seed sourcing.
Yeah, so that probably relates to this next question from Jenny Wong, who says, how do your results relate to the identification of GCUs for these species?
Again, so this is something that probably we be able to answer better in the next stage of the project. We are not yet there. We found some genes that could be under selective pressure in forestation protocols, and some of them can be relevant for even ecological adaptation or local adaptation, but we don't know for sure yet.